So in the last lecture, we discussed the ways in which synaptic transmission occurs. We discussed exocytosis, we talked about receptor activation, the way neurotransmitters are synthesized, as well as what happens to the neurotransmitters to remove them from the synaptic cleft to inactivate them. We're going to be spending this particular lecture talking about the different kinds of neurotransmitters that exist within the nervous system, and we're going to end the lecture discussing the ways in which psychoactive drugs kind of utilize this natural neurotransmitter function to alter psychoactive states of human beings. So if you're a coffee drinker, you're a drug user. And as somebody who loves coffee and caffeine and is definitely addicted to it, caffeine is a drug. People are addicted to it. They derive a high from it, even if that means waking up. And this is one of the many different ways in which human beings utilize drugs. Granted, caffeine isn't in the same category as some of the, harsher or more addictive drugs like methamphetamine or heroin, but it certainly is a drug because it alters the natural state of some of the chemicals that exist within our nervous system. And later on in the lecture, we'll talk about the ways in which caffeine works within the nervous system to produce your more alert, awake state that you might experience if you drink coffee or soda or anything with caffeine in it. So this is going to be the lecture this is going to be the outline for the lecture. We're going to first talk about the different neurotransmitters that exist. We'll talk about the classic neurotransmitters that we discussed in the previous lecture, the small ones that are made in the terminal, and then we'll transition into the neuropeptides. We're not going to be spending much time on the neuropeptides other than to kind of reiterate what you already know, but that's to distinguish them from the classic neurotransmitters, which many of the drugs out there actually target. They target the classic neurotransmitter action within the brain. Then we'll talk about the pharmacology of synaptic transmission, the ways in which drugs can affect neurotransmitter action to produce the different symptoms or feelings or highs from taking the drug. And lastly, we'll discuss a couple of psychoactive drugs and the way in which it affects the pathway, the reward pathway within the brain to produce addiction. So these are the categories of neurotransmitters that we'll be discussing in this lecture. This is the table from your textbook, so it's no different. We'll be spending a good amount of time on the monoamines as well as the amino acids. We won't spend too much time on the peptide neurotransmitters, but we will be touching on acetylcholine. Some of the neurotransmitters, such as anandamide, which is actually a THC-like substance, so it acts on the same receptors that THC, the active substance in marijuana, works on, but it's made within the brain as a neurotransmitter. There's also adenosine, which is what caffeine blocks, and again, we'll touch on that later in this lecture. So this is kind of how we're going to get started. We're breaking down the neurotransmitters into their different categories. So the larger categories are listed on the left. We've got amino acid neurotransmitters, monoamines, acetylcholine, which gets its own very special category, and then neuropeptides. And then we'll talk about the specific neurotransmitters within each category. So let's get started with amino acid neurotransmitters. These are the most prevalent, most common neurotransmitters. Actually, most of the work that's done in the nervous system is the result of these two specific neurotransmitters, glutamate and GABA. So glutamate is the major excitatory amino acid neurotransmitter. What this means is that whenever glutamate binds to its receptor, it almost always produces an EPSP. It depolarizes the membrane and makes it more likely that an action potential will occur. GABA, on the other hand, is the major inhibitory amino acid neurotransmitter. Just like glutamate excites consistently, GABA inhibits. So when GABA activates its receptor, it almost always produces an IPSP, or a hyperpolarization. It makes the membrane potential more negative and less likely that an action potential will occur. So as I mentioned earlier, almost every single central nervous system synaptic transmission is accomplished through the actions of one of these two neurotransmitters. So let's talk about glutamate first, as I promised in the previous slide but didn't actually do. So glutamate is synthesized from an amino acid. So if you're not familiar with what amino acids are, they are the building blocks of proteins. So glutamine is a type of amino acid, and the enzyme glutaminase takes glutamine and converts it into the active neurotransmitter glutamate. Glutamate is then packaged into vesicles, released through exocytosis at the synaptic cleft, and then activates 4 different types of receptors. 3 of them are ionotropic. The first one is AMPA, the second one is kainate, and the third one is NMDA. All 3 of them are ionotropic. If you remember from the previous lecture, ionotropic receptors are ion channels. Once the neurotransmitter binds, they just open up the channel, and then once the neurotransmitter leaves, the channel closes. The NMDA receptor is a really unique receptor, and we'll talk about it when we get to talking about learning and memory later on in the semester. But all of these receptors need glutamate to open them, and they pretty much allow positive ions into the cell. So I hope you guess that it's sodium that it lets in, and that sodium entering the cell ends up producing EPSPs. There's a metabotropic glutamate receptor that also activates glutamate. So remember, metabotropic receptors are those that have a G protein attached to it and can generate second messengers and produce a whole host of different things. But the vast majority of times, this metabotropic receptor ends up producing an EPSP in the postsynaptic cell. Let's talk about GABA. Ironically, glutamate serves as the building block for GABA. So glutamate is what we call a precursor for GABA. So it's ironic because glutamate is the excitatory neurotransmitter, and we use it to make the inhibitory neurotransmitter GABA. So GABA stands for gamma aminobutyric acid. You will not need to know that, but in case you are curious, GABA stands for gamma aminobutyric acid, and it's converted from glutamate by the enzyme GAD, glutamic acid decarboxylase. Just like glutamate had multiple receptors, GABA does as well. All of them produce IPSPs, so they inhibit the nervous system. They hyperpolarize the membrane potential. There are 2 large categories of receptors. You have GABA A, which is ionotropic, and GABA B, which is metabotropic. So the way in which alcohol and benzodiazepines, for example, work to sedate or inhibit the nervous system is through the actions of the GABA A receptor. So what they actually do is they keep the GABA A receptor open for a lot longer than if GABA was there by itself. So the way in which alcohol works to sedate or inhibit the nervous system is through the actions of the neurotransmitter GABA, the major inhibitory neurotransmitter GABA. All right, we're going to move on to monoamines. There are 2 categories of monoamines. We've got catecholamines and indolamines. We're going to discuss the catecholamines first. So within catecholamines, we have dopamine, epinephrine, and norepinephrine. So the reason why these classic neurotransmitters are called monoamines is because they have a chemical group that's called an amine, and they only have one of it, which is why they're called monoamine. So the way in which catecholamines are produced are actually all in one pathway. They're produced from the amino acid tyrosine. So tyrosine we get from our diet. Again, just like glutamine is an amino acid, tyrosine is also an amino acid. It's typically used to make other proteins, but it can also be used by the nervous system to make dopamine, norepinephrine, and epinephrine. So tyrosine from our diet is converted by the enzyme tyrosine hydroxylase to an intermediary chemical called L-DOPA. L-DOPA is converted by enzyme 1, you don't need to worry about it, by enzyme 1 into dopamine. If the neuron releases dopamine as its neurotransmitter, the process would stop here. However, if the neuron were to be making norepinephrine, dopamine would further be converted by another enzyme into norepinephrine. And again, if the neuron is making norepinephrine as its neurotransmitter, it would stop here. But if it was making epinephrine, norepinephrine would further get converted by a different enzyme into epinephrine, which would then be released as the neurotransmitter. So just keep in mind that depending on the kind of neuron it is and what neurotransmitter it makes, the process could stop here if it's a dopamine-producing neuron. It could stop here, so dopamine would be made, but it would be converted into norepinephrine if it was a norepinephrine-producing neuron. And similarly, if it was an epinephrine-producing neuron, it would continue, norepinephrine would be further converted into epinephrine. So you might not be aware of this. You've probably heard of the term adrenaline. Well, epinephrine is a fancier term for adrenaline. It's one and the same. So epinephrine and norepinephrine are produced by the adrenal gland, but they are also used as neurotransmitters within the nervous system. So dopamine, and actually all catecholamines, are classic neurotransmitters, so they're made in the presynaptic terminal. They are packaged into vesicles and released through exocytosis, just like most neurotransmitters are. They bind to dopamine receptors in the postsynaptic cell, and the vast majority of dopamine receptors are metabotropic, and we'll talk about them in a second. It's removed from the terminal, so dopamine and norepinephrine and epinephrine are all removed from the terminal using the transporter mechanism, and there's a transporter, reuptake transporter for each neurotransmitter. Sadly, it's again not very creative, but the dopamine reuptake transporter is known as DAT. The norepinephrine transporter is known as NET. And so the dopamine, norepinephrine neurotransmitters are removed from the synaptic cleft through the reuptake mechanism. They can be broken down by an enzyme called MAO, monoamine oxidase, into component parts, which can then be recycled, or dopamine itself can be recycled and repackaged for release. Again, I'm only using dopamine as an example, but this can be applied to all catecholamines, norepinephrine as well as epinephrine. So dopamine has 5 different receptor subtypes that are all metabotropic. So just to remind you, they are G protein coupled receptors that utilize second messengers. Dopamine as a neurotransmitter is utilized in many different functions within the nervous system. Movement, which is affected in Parkinson's disease. Attention, many ADHD drugs affect dopamine as well as norepinephrine. Learning, as well as something we'll talk about at the end of this lecture, the reward pathway. The vast majority of drugs that are addictive are addictive because they increase dopamine within the reward pathway, giving us the feeling of pleasure. So dopamine action is responsible for the reinforcing effects of drugs. Dopamine, again, just to kind of touch on what you know right now. So dopamine is reduced in Parkinson's disease, right? So the cells that make dopamine die off in Parkinson's disease. So what do? They administer L-DOPA. If you remember, L-DOPA is that intermediary chemical before dopamine is made. So the reason why L-DOPA is administered instead of dopamine is because it doesn't cross the blood-brain barrier. Dopamine doesn't cross the blood-brain barrier, but L-DOPA does. So L-DOPA relieves Parkinson's disease symptoms by allowing the cells that are still alive that make dopamine to make more dopamine. Interestingly enough, there's too much dopamine activity in parts of the brain in schizophrenia. So what do they do to treat schizophrenia? They give a treatment that blocks dopamine receptors. So when you think about where dopamine works within the brain, when we talk about the side effects of these treatments, it should come as no surprise. Administering L-DOPA in large enough doses actually produces psychotic-like symptoms. And interestingly enough, a side effect of administering antipsychotics, which are drugs that block dopamine receptors, is you produce Parkinson-like symptoms. And it's actually, there's a term for it called Parkinsonism. But again, knowledge is power here. If you know where these neurotransmitters are acting within the nervous system, you might be able to predict the side effects that result from administering or changing the action of these drugs within the brain. Norepinephrine is a catecholamine as well, like dopamine. It's involved in attention and arousal as well as feeding behavior. It's part of sleeping and dreaming. Epinephrine, as we talked about, is also known as adrenaline. It seems to be involved in eating as well as many visceral activities. So things like, it's part of the stress response. Both of these neurotransmitters, bind to what are known as adrenergic receptors. They have different categories of adrenergic receptors, but they all belong to one class. All adrenergic receptors are metabotropic. They utilize G protein and second messengers. All right, let's move on to monoamines again, this time talking about indolamines. And the only indolamine we're going to worry about today is serotonin. So many of you are probably familiar with serotonin because the drugs that relieve depression, a very common mental illness, utilize or do it through modifying serotonin action within the brain. So again, just like catecholamines, serotonin has a single amine group. Serotonin is produced by tryptophan, which is an amino acid. It's converted to 5-hydroxytryptophan, or 5-HTP. You can abbreviate it 5-HTP. And then 5-HTP is converted by another enzyme into serotonin, which is chemically known as 5-hydroxytryptamine, or 5-HT. So you might find that I abbreviate serotonin as 5-HT because it's a lot quicker to write 5-HT than it is to write serotonin. So serotonin is involved in so many different functions within the nervous system. It's most well known for its actions in mood, specifically depression. It also is part of regulating emotion as well as sleep. Serotonin transporters are utilized as antidepressants. So as I mentioned earlier, catecholamines are removed from the synaptic cleft using reuptake transporters. Serotonin is also removed from the synaptic cleft using reuptake transporter, except that it's known as the SERT, serotonin reuptake transporter. Antidepressants work to increase levels of serotonin at the synapse by blocking this reuptake transporter. A common SSRI is fluoxetine or Prozac. So just to illustrate to you how antidepressants work, serotonin is removed from the synaptic cleft through a reuptake transporter known as SERT. SSRIs, selective serotonin reuptake inhibitors, block this reuptake transporter and therefore increase the level of serotonin in the synapse, because serotonin has no way to get out of the synapse into the cell, so more serotonin hangs around in the synapse and continues to activate its receptor. And this is one of the modes of how it relieves depression. Serotonin, just like dopamine, is also broken down by monoamine oxidase once it enters the cell. To date, there are at least 17 known subtypes of serotonin. The vast majority of these are metabotropic, but there is one ionotropic receptor that serotonin acts on. Acetylcholine gets its own category of neurotransmitters, and the way in which acetylcholine is made, again, is by products from our diet. So choline and acetyl coenzyme A are the two components needed to make acetylcholine. And interestingly enough, it's made by an enzyme called choline acetyltransferase, or CHAT. It's sometimes abbreviated CHAT. It transfers acetylcholine transfers the acetyl onto choline. Again, not very creative. And so you make acetylcholine. You'll notice that I abbreviate acetylcholine, ACH, because it's a very long neurotransmitter to write out. Acetylcholine is packaged into vesicles, as other classic neurotransmitters are, which are also made in the terminal. It's released through exocytosis and binds and activates receptors on the postsynaptic cell. But unlike monoamines, acetylcholine is broken down through enzyme degradation by an enzyme known as acetylcholinesterase, or ACE. It's broken down into choline and acetate, and choline is transported back into the terminal through a transporter to be recycled and reused to make more acetylcholine. Acetate or acetic acid just floods into the extracellular membranes, extracellular fluid, and then eventually enters the bloodstream and is excreted out through the urine. So this is your book's representation of how acetylcholine is made. Choline and acetyl coenzyme A kind of come together to make acetylcholine. And again, you don't need to worry about the chemical structure, but here it is in case you are curious. And here are some foods that most commonly have choline in them so that we can make more acetylcholine. There are two different categories of acetylcholine receptors. They're nicotinic and muscarinic. Nicotinic, which shouldn't surprise you, is the receptor that can be activated by nicotine, which is part of the reason why it got its name. And there is a chemical in a mushroom known as muscarine, which activates muscarinic receptors. Both of these receptors utilize acetylcholine as their neurotransmitter. Nicotinic receptors are ionotropic. Muscarinic receptors are metabotropic. All muscle movements in your body, whether it's skeletal muscles or smooth muscles, utilize nicotinic receptors. You can imagine why it would be important to utilize an ionotropic receptor for especially skeletal muscle movements. You want these things to occur very, very quickly, and so all skeletal muscle movements occur through nicotinic receptors. But acetylcholine is also utilized in the autonomic nervous system, and it regulates the parasympathetic functions all over the body. It's also important in REM sleep functioning as well as in learning and memory. So many of the drugs utilized to treat Alzheimer's disease work through to alter acetylcholine function. So it is thought that acetylcholine seems to play a role in cognitive functions such as learning and memory. Our last category of neurotransmitters are neuropeptides. There are hundreds of neuropeptides, but endorphins, which you may have heard of, is a type of neuropeptide. Like all large neurotransmitters, these neuropeptides are made through the process of transcription and translation. DNA is utilized as the template to make the single-stranded mRNA. Ribosomes take this mRNA and make what's called a precursor protein that is much larger, and then this precursor protein is chopped up into the active neuropeptide or peptide neurotransmitter. But then it's packaged up by the Golgi apparatus and sent down to the terminal to be released. So the process of making large neurotransmitters is much more time intensive than making small neurotransmitters, but some examples of neuropeptides are endorphins that we talked about. These endorphins work like endogenous opiates. Endogenous meaning made from within. Opiates are like heroin and morphine, but these are produced at much, much lower levels, and we're able to, but they produce similar effects. Oxytocin is also a neuropeptide that's involved in childbirth as well as nursing or lactation, but it's also thought to be the neuropeptide that allows for bonding between either mother and child or between, partners. Corticotropin releasing factor is an anxiety-producing neuropeptide. It's thought to be involved in the process of stress as well as anxious behavior. The neuropeptides, based on what they are and where they're produced, have a variety of functions within the nervous system. We're only going to talk about them as they come up, but most peptide receptors are metabotropic. Let's talk about something that's probably more interesting for you, and that's how drugs may affect neurotransmitter function. So now that you kind of know the major classes of drugs within the nervous system and the ways in which synaptic transmission occurs, you might be surprised to find that you can figure out what a drug may do based on the information that's provided in kind of the description of the drug. So drugs can be administered a whole host of different ways. Here are some listed in your textbook. It can be administered through oral, through injection, inhalation. The route of administration is going to dictate how quickly it has its effects in the central nervous system. But then it also needs to be absorbed. So if it's administered orally, it goes through the digestive tract, and then it needs to be broken down by the liver and may enter storage sites like fat and bone. Some of it may get excreted, but some of it gets absorbed. So the dose of the drug depends largely on how it's administered, as well as a variety of things like how tightly it binds to the receptor, to its binding sites. So if you're thinking about going into psychopharmacology, for example, those kinds of pieces of information are going to help you better design drugs to treat various illnesses. The other aspect of designing drugs is what's known as the therapeutic index. Every single drug out there, from aspirin all the way to ones that are really highly regulated, like oxycodone, have what are known as therapeutic indices. There is, so what you're looking at here is what's called a sigmoidal curve. Every drug takes on this kind of shape where its effects at lower doses are smaller and at higher doses are higher, but there's kind of a sweet spot where you kind of want to be in terms of dose administration. You don't want to be too high where you kind of lose the effectiveness of the drug. But regardless, shown here in green is the therapeutic effects of the drug, and shown here in black are what's known as the toxic effects of the drug, those effects of the drug that will eventually kill you. So this is the dose of morphine. Morphine can produce pain relief if administered in quantities that are therapeutic, but if administered at large enough doses, you can actually kill the person and affect things like their ability to breathe. You'll notice that there's overlap between the dangerous effects and therapeutic effects of the drug, right? This is the same exact dosage shown here. What you want to do is you want to stay within the safe zone where the vast majority of the population experiences therapeutic effects and not dangerous effects. So every drug has what's called a therapeutic index, and the therapeutic index, you want it to be large between the therapeutic dose and the dangerous toxic doses. So the larger the spread between the therapeutic and dangerous doses, the better the therapeutic index for the particular drug we're talking about. So morphine, as you can see, has a very low therapeutic index. Small amounts of it, just doubling a small dose, could potentially kill a human being. So in terms of drug action, let's talk about some of the ways in which neurotransmitter action is affected by drugs. We're going to be kind of diving into the level of the synapse to discuss the ways in which drugs can affect nervous system function. Before we do that, there is an aspect of synaptic transmission that we did not discuss that I'd like to talk about here. So I wanted to introduce you to the concept of autoreceptors. Whoops, there it is, the concept of autoreceptors. So autoreceptors are found on the presynaptic terminal, shown here. They bind to the neurotransmitter that this neuron releases, and their sole function is to regulate the neuron's release of neurotransmitter. So a neuron has no clue how much or how little neurotransmitter it's releasing, but through the activation of autoreceptors, it gets a clue as to how much neurotransmitter is being released. So if the autoreceptors get activated, it tells the neuron, oh, you're releasing too many neurotransmitters, you got to dial back. So the autoreceptors serve as a way for the neuron to monitor neurotransmitter release, and the activation of autoreceptors almost always results in the reduction of neurotransmitter release for the future. We'll talk about the ways in which autoreceptors are affected by drugs and what that does to neurotransmitter action later on, so don't worry about some of the content in this particular slide. Okay, so there are 6 steps in neurotransmitter action. We've got synthesis, right? Enzymes take the precursors to make neurotransmitters. We've got storage. Neurotransmitters need to be packaged into vesicles, and they're typically pumped into these vesicles. They are released through exocytosis, which is step 3. They bind to postsynaptic receptors, which is step 4. They can bind to autoreceptors, which is going to affect neurotransmitter production and release. They can also be broken down through either reuptake or enzyme degradation. So it is through these 6 steps that most drugs produce their effects on the central nervous system. And let's talk about the ways in which it can do that. But first, a drug can affect the function of the nervous system in one of 2 ways. If it increases neurotransmitter action, we call it an agonist. If it decreases neurotransmitter action, so it prevents the neurotransmitter from doing what it normally does, it's considered an antagonist. So let's put this into play in the 6 different steps that we talked about in the previous slide. Oh, right. So any of the 6 steps can be modified to achieve agonistic or antagonistic effects. So let's take a look at them. So if drugs increase the neurotransmitter precursor, as is the case with L-DOPA, the drug is considered an agonist. If a drug destroys or blocks synthesizing enzymes, the neurotransmitter won't be made, and so it will be decreasing neurotransmitter action, and the drug will be an antagonist. So I'd like you to not look at your notes if you have them printed out and try to guess whether a drug could be an agonist or antagonist based on what I describe here. So if a drug prevents the storage of neurotransmitter in vesicles, would it be an agonist or an antagonist? If you said an antagonist, you are right, because neurotransmitters can only be released if they're in vesicles, and if the drug prevents these neurotransmitters from getting into the vesicles, they won't be released through exocytosis. How about stimulating the release of neurotransmitters by opening up the calcium channels? If you said agonist, you're right, because it increases neurotransmitters into the synaptic cleft. How about decreasing neurotransmitter release by perhaps blocking the calcium channels? It would be an antagonist. Stimulating postsynaptic receptors. It would be an agonist, because it would be doing exactly what the neurotransmitter typically does, except that this is a drug. If it blocks postsynaptic receptors, it would be an antagonist, because it's preventing the neurotransmitter from doing what it should be doing, which is activating postsynaptic receptors. How about stimulating autoreceptors? So keep in mind, when you activate an autoreceptor, it's going to tell the neuron you're making too much neurotransmitter, and the function of the autoreceptor is to decrease neurotransmitter function. So if you stimulate the autoreceptor, is it an agonist or an antagonist? If you said antagonist, you are right, because it's preventing the neurotransmitter from being produced and released. Conversely, if you block the autoreceptor, it would be an agonist, because the neuron should be told that it's making too much, but it's not getting the message because the autoreceptor is blocked, and so the drug is an agonist. If you block reuptake, as was the case with SSRIs, it would be an agonist. And then if you block degrading enzymes, the ones that are responsible for breaking down neurotransmitters, it would also be an agonist. So hopefully you can see the ways in which drugs, by acting in these 6 different steps, can modify neurotransmitter action and ultimately affect nervous system function. So this is your textbook that kind of demonstrates these 6 steps a little bit differently than the way we talked about, but it effectively is getting at the same idea, that at the synaptic transmission level, drugs can modify nervous system function. So let's talk about caffeine, right? I promised you you would learn the way in which caffeine works to wake you up or kind of stimulate you. So caffeine is a stimulant, and we'll talk about the different classes of drugs in a second, but the way in which caffeine works is actually by antagonizing or blocking a receptor. So let's take a look at this. So there is a neurotransmitter known as adenosine. Adenosine, when it activates its receptor, inhibits the neuron, so generates an IPSP. So when adenosine levels are high, we feel sleepy. What caffeine does is it blocks the adenosine receptor, preventing adenosine from activating its receptor and therefore stimulating the nervous system. So caffeine is actually an antagonist for adenosine. But because adenosine is largely inhibitory, the effects of caffeine are to stimulate the nervous system and wake us up. Dopamine works a little bit differently. Cocaine works a little bit differently. The way in which it increases dopamine levels at the synapse is by blocking the dopamine reuptake transporter, or DAT. By blocking dopamine reuptake transporters, you're going to prevent dopamine from being removed from the synaptic cleft, and it's going to continue to activate dopamine receptors. So remember, one of the actions of dopamine in the nervous system is movement, and many people who end up abusing cocaine or even amphetamine, which works on dopamine, often can't sit still and are constantly moving and getting involved in many, many different things. So knowing the way in which the drugs work can help you to determine the action the drug is going to have on the nervous system and ultimately behavior. So there are many different categories of psychoactive drugs. We've got CNS stimulants. So some stimulants are amphetamine, cocaine, caffeine, as well as nicotine. CNS depressants, such as alcohol, barbiturates, and benzodiazepine, they all largely work through the actions of GABA. We have analgesics, the pain relievers, like opium, morphine, and codeine. We have hallucinogens that we're not going to necessarily talk about, such as mescaline, LSD, and psilocybin, as well as drugs with diverse CNS effects, such as marijuana, and then various drugs that are used to actually treat mental illness, which are known as psychotherapeutics. So Prozac and Thorazine, which are used to treat depression and schizophrenia, respectively, would fall into this category and not any of the above-mentioned categories. So I mentioned before that drugs of abuse have their addictive properties through the release of dopamine in the reward pathway. So I wanted to talk to you a little bit about what that reward pathway is. So the reward pathway is known as the mesolimbic dopamine pathway. So the meso, standing for midbrain, involves the ventral tegmental area. The ventral tegmental area projects and releases dopamine onto the nucleus accumbens. This mesolimbic pathway is also known as the reward pathway, and what drugs of abuse do is they release dopamine to such high levels within this pathway that no natural reward could compare. And so the feeling of pleasure or high derived from the activation of dopamine within this pathway is so addictive that the person often seeks out that feeling again, and repeated abuse of the drug ensues. What's interesting is that over time, the brain is going to compensate. It is a very, very smart, efficient organ. Over time, the brain is going to compensate for this drug being on board, so it's going to try to counteract the effects of too much dopamine within the brain. So you'll notice that one of the things that dopamine does from the VTA is it stimulates the cortex as part of the mesocortical pathway. So when the drugs of abuse stimulate ventral tegmental area activation, it causes dopamine release in both the nucleus accumbens and the prefrontal cortex. But too much stimulation of dopamine over time actually results in what is known as hypofrontality. The normal steady state of activation of the prefrontal cortex in a chronic drug abuser is actually underactive than what it should be. So many individuals who are chronically abusing cocaine may see brain activation that looks like this. So when we check for glucose metabolism through PET scan, remember we talked about PET, positron emission tomography, a couple of lectures ago, we can measure the activity of brain areas, and the more red we see in the brain, the more active that area is, and the more blues and greens we see, the less active the area is. In somebody who's addicted to cocaine, you can see that the whole cortex, which is shown here, is highly, highly underactive in comparison to somebody who may have been administering saline. And so your brain on drugs is not something like it's overstimulated in a frying pan, but it's more like something in a refrigerator where it's very, very cooled down and shut down and underactive. So chronic drug use can actually underactivate or cause underactivation of brain areas, counteractive to what you might think, which is that, chronic drug use might be overstimulation. So let's summarize this lecture. There are several different types of neurotransmitters that we talked about. We talked about amino acid neurotransmitters, which I called amines here, but that should be amino acid neurotransmitters. We've got monoamines, which include the catecholamines and indolamines. We've got acetylcholine as well as neuropeptides. Each neurotransmitter has a variety of receptors known as subtypes onto which they act to control different functions within the nervous system. Drugs that affect the nervous system typically act at the level of the synapse at one of the 6 steps of the neurotransmitter action that we talked about. And depending on the drug's effects at one of these 6 steps, it can serve either as an agonist or an antagonist for that neurotransmitter. There are several different categories of psychoactive drugs that we discussed, but the drugs that have addictive properties get their addictive properties from acting on the reward pathway, the mesolimbic pathway from the ventral tegmental area to the nucleus accumbens that utilizes dopamine as its major neurotransmitter. So that's it for this lecture. Don't forget to submit your reflection.